Even though modern fluorescence microscopy produces detailed three-dimensional datasets, colocalization is still most commonly investigated in two dimensions. Since region of interest selections are also made two-dimensionally, complex three-dimensional structures cannot be adequately isolated. Therefore, in this video we will demonstrate a virtual reality system that is capable of complex three-dimensional region of interest selections. Firstly, the system visualizes the three-dimensional reconstruction of the fluorescence data. In this case, a neuronal cell is shown. The sample can be transformed and navigated in three dimensions using a gamepad. Furthermore, several visualization parameters can be changed easily, such as a noise filter and the sample's opacity. A maximum intensity projection can also be visualized. Secondly, we can visualize colocalization in various ways, such as only showing the colocalized voxels in their fluorescent colors, or in white, or overlaying these voxels on the rest of the sample. Region of interest selectivity in two dimensions remains limited, and existing tools do not allow precise sample navigation to accurately position these selections. With the virtual reality guided system, we can make precise selections using a variety of selection tools. More complex selections can be made by using a combination of the available tools. Here we isolate the perinuclear region by using two cylindrical selections. Once a selection has been made, the selection can be isolated from the rest of the sample and be visualized by itself, thereby improving the navigation through the sample. As with these cylindrical selections, in two dimensions it is not possible to make selections that adequately describe the complex three-dimensional structures. These selections can however easily be converted to three-dimensional ellipsoidal selections. Note that the selections can be scaled along any axis and rotated around the z-axis. In virtual reality, selections can easily be verified, as we scale the two internal ellipsoids to only select the perinuclear regions. Selections such as these cannot be made in two dimensions, and since more voxels are taken into account in the three-dimensional selections, there is also more statistical confidence in the colocalization analysis results. Here we demonstrate selecting the middle region with several cylindrical selections. As more selections are added, they retain the size of the previous selection. These selections can now be translated and scaled either independently or simultaneously, as is demonstrated here. This allows consistent sampling of certain regions within the cell, as well as between different cells. Again, these selections can easily be converted to ellipsoidal selections. The virtual reality system further improves the navigation through the cell by allowing a specific region of the sample to be enlarged for more accurate subsequent selections. In this example, we use the Freehand tool to select some microtubulin strands. These strands can be visualized and analyzed isolated from the rest of the sample. Lastly, after the region of interest have been selected, several colocalization metrics and scatter plots can be calculated within these regions. In summary, in this video we have demonstrated the use of virtual reality-enabled interactive three-dimensional region of interest selection to rapidly isolate several noteworthy cell features to visualize and analyze colocalization in these selections. The improvements that three-dimensional selections offer over the standard two-dimensional selections are further explored in the paper. It is hoped that future analysis of samples of biological relevance can be interrogated with greater precision and navigation control, thereby fully exploiting the potential of fluorescence-based image analysis in biomedical research. Thank you for watching.